Hi, good morning and good afternoon, everyone. My name is Eleanor Cowley, and I'm a Senior Automation FAS for Roche Sequencing. And today I'll be talking about automating RNA-seq library preparation using standard automated liquid handling devices. So here's a quick overview on what will be presented during today's webinar. First, we will review some benefits of automation and why you may want to consider automating your NGS workflows. I'll also share some general tips for developing automated workflows. Then I'll discuss the automated RNA-seq library preparation kits that are offered by Roche and provide an overview of our Roche Sequencing automation program. We'll then dive into what our automation program has to offer, including our approach to method development, and I'll share a bit about how our team demonstrates and assesses automated method performance. And lastly, we will end by reviewing examples of automated KAPA RNA HyperPrep workflows across 3 different automated liquid handling platforms. And those include the PerkinElmer Sciclone, the Tecan Freedom Evo, and the Beckman Coulter Biomek i7. So let's jump right in. Next generation sequencing technology is evolving rapidly, and it's now cheaper and faster than ever before. And at the same time, the demand for high throughput workflows has drastically increased. Automating NGS workflows has some very clear benefits, and this starts with scalability. So once you've developed an automated library prep workflow in a liquid handler, you can greatly increase your throughput to process larger numbers of NGS samples. So for example, in the same time it would take an individual to manually process 16 samples, many of these robotic platforms can process up to 96 samples. In addition, some platforms offer multi-plate processing, and this can drastically increase your throughput even more. So next up, we have consistency. And although library prep workflows have improved over the last few years, manual library prep methods can still be monotonous and labor intensive, leaving them vulnerable to human error. In addition, manual sample handling is prone to run-to-run variability. So in contrast, a well-designed, optimized, automated NGS workflow decreases the number of manual steps and minimizes the risk for human error and variability, which leads to the production of consistent, high-quality data. Of course, automating your NGS workflows also has the added benefit of increased walkaway time. And although this benefit varies depending on the automation platform, at the very least, users will no longer have to spend time performing repetitive pipetting tasks at the bench, and they can focus that time elsewhere. And lastly, automation of NGS workflows can help drive innovation. So by eliminating bottlenecks, scaling up of NGS workflows is easier than ever before. And this increased throughput allows labs to address novel research questions that might not be feasible if they were relying solely on manual processing. So some examples include drug discovery, microbiomics, plant breeding, disease research and diagnostics, and even environmental monitoring. And each of these application areas requires screening of large numbers of samples using NGS. An increased innovation can also mean that you have more time to spend on collaborations, opening doors for new projects and scientific discoveries. So now that we've summarized some of the immense benefits of automation, we'll talk about the process of developing a robust, well-designed automated method. This process can be quite challenging, but our Roche Sequencing automation team has a great deal of experience creating automated NGS workflows. So in the following slide, I'll share a few tips and tricks to help you focus on what's most important when you're looking to automate your NGS workflows. So first, when developing an automated NGS workflow, we would strongly recommend investing the time upfront on the optimization process. This gives you a much better shot at successful method performance in the first few rounds of live testing, minimizing the amount of troubleshooting at the later stages of the development process and saving time in the long run. Most NGS workflows involve viscous enzymatic master mixes that are much more difficult to transfer than aqueous solutions. So to account for this, our team spends the majority of method development time on optimizing liquid transfer steps, ensuring that accurate volumes are delivered throughout the entire workflow. One of our tricks is to use reagents that are as close as possible to the actual workflow reagents, and that would include expired, non-viable reagents. It's also important to pay close attention to bead cleanups in order to minimize or eliminate bead carryover and or loss, especially during those supernatant removals. So next, when designing an automated NGS workflow, make sure you understand the limitations of your system that you plan to use. Many of these workflows require accurate pipetting of small volumes, so you'll want to be sure to choose an instrument that's designed to work within the volume range of your assay. In addition, select consumables that are appropriate for the assay's volume range. So depending on the consumable that's selected, reagents may require more or less dead volume in order to ensure accurate transfer of the desired volume to the selected number of wells. And since NGS reagents are not cheap, consider these dead volumes during development to avoid wasting expensive reagents whenever possible. And as a note, all of Roche's 96 reaction NGS library prep kits are actually designed with automation in mind, providing enough dead volume to allow multiple automated runs with the methods that are developed by the Roche Sequencing automation team and even our vendor collaborators. So our third recommendation would be to pay close attention to temperature specifications that are provided in the workflow's technical data sheet or user's guide. And this includes both heating and cooling steps. So for example, if you do plan to perform incubations on deck using open air Peltier devices, make sure that your device is able to get the sample wells to the correct temperature. Many times these open air Peltier devices need to be offset by 2 to 3 degrees in order to compensate for heat dissipation. And in addition, when automating methods with extended high temperature on deck incubations, we recommend using a synthetic oil overlay in order to prevent evaporation. We've had success with QIAGEN's VaporLock as well as Sigma Aldrich's silicone oil. When using these oil overlays, we typically add the oil to the reaction just prior to the incubation, and we would remove it later in the workflow with a bead cleanup step. Sample mixing is also extremely important for almost all reactions in a standard NGS workflow, and mixing can be done either by standard up and down tip mixing or with an integrated on-deck shaking device when available. For more difficult to resuspend wells, some systems actually allow users to tip mix while the plate is shaking, and this makes it easier and faster to homogenize reactions. If you are using an integrated shaking device, it's important to make sure that the reaction volumes and the shaking speed are optimized in order to avoid cross-contamination of wells. And lastly, I want to highlight that the Roche Sequencing automation team has a wide range of expertise automating NGS workflows across a number of different platforms. And we're happy to offer help where needed to support you in automating any of our kits and workflows. So now let's talk about automatable RNA-seq library prep kits and what Roche has to offer. And today, I'll mainly focus on our single-day streamlined strand-specific workflow offering, which is called KAPA RNA HyperPrep. The core library prep workflow is shown on the left side of the screen. It takes about 4 hours to complete. It starts with fragmentation and proceeds through first strand synthesis, a combined second strand synthesis and A-tailing reaction, adapter ligation, 2 bead cleanup steps, library amplification, and a final bead cleanup step. This workflow is compatible with 3 optional upfront enrichment modules. The first is mRNA capture, in which polyadenylated transcripts are selected for over other RNA species, including ribosomal RNA, leaving mature mRNA for subsequent downstream library preparation and sequencing. The second enrichment option is our KAPA riboRase kit, in which ribosomal RNA is directly removed from the total RNA sample, allowing for the sequencing of remaining species, which includes mRNA, as well as non-coding and precursor transcripts. The third enrichment option is our KAPA riboRase with globin depletion kit, which is optimized for use with blood-derived RNA samples. In this workflow, globin transcripts are directly removed along with ribosomal RNA prior to library preparation. And both of our riboRase kit offerings are compatible with human, mouse, and rat samples. If you wish to deplete the ribosomal RNA from any other sample type, such as bacterial or plant samples, we now offer an online probe design resource called KAPA Ribo Designer for custom probe design. So our total KAPA RNA HyperPrep workflow without enrichment and our KAPA riboRase kit offerings are suitable for use with degraded inputs and are amenable to lower inputs compared to other commercially available kit offerings. So our total KAPA RNA HyperPrep workflow requires a minimum input of 1 nanogram of total RNA, while our KAPA riboRase workflow requires a minimum input of 25 nanograms. All of our RNA-seq library prep kits do include automation-friendly workflows and kit configurations, and the 96 reaction kits provide adequate overages to meet minimum dead volume requirements for most automated liquid handling platforms. And so now that we've discussed what Roche has to offer in terms of automatable RNA-seq library prep kits, let's take a look at what automated methods are currently available for these workflows across some common liquid handling platforms. So our automation program develops standardized solutions for Roche NGS workflows on several third-party liquid handling platforms. The vendors and the instruments that we currently support are shown down the left side of the table. And for each of these, we have either currently available methods or methods that are in development. Across the top of the table, you'll see the list of automated Roche NGS workflows, including library preparation for both RNA and DNA, library QC, target enrichment, and oncology workflows. The blue box highlights available automated solutions for Roche's RNA HyperPrep kits. And with each of the vendors that are shown, we've built collaborative partnerships where we focus on developing standardized solutions. And this means that we generally develop our applications on NGS-specific deck layouts if the vendor has one. These methods are usually mid to high throughput depending on the platform, and they're designed to be both flexible and modular to suit a wide range of users' needs. Our team of experts offers both consultative as well as on-site support to assist users in automating their NGS workflows. So when it comes to developing new automated solutions, we take both internal and collaborative approaches. In some cases, our team will independently develop methods on platforms that we have in our applications lab. And in other cases, we will partner with vendors to develop methods on their platforms for our customers. So we have a number of platforms that are available in our own lab. And to name a few, we have the Hamilton NGS Star, a Tecan Freedom Evo NGS Workstation, an Agilent Bravo NGS Workstation, and Beckman Coulter's Biomek FXP and Biomek i7 platforms. Of course, the space in our lab and our resources are limited, which is why we have worked really hard to build partnerships with each of the major vendors to be able to do collaborative development on the platforms that we don't have in our lab. So with either of these approaches, we're able to develop and demonstrate method performance, keep track of versioning, and produce documentation. And the documentation includes both application notes and technical notes that feature our development work. So another important focus for our team is customer consultation and support, which we do provide free of charge to our customers, in addition to providing standard email and phone support to address questions about available automated solutions. We also provide on-site and remote support, traveling on-site to install methods, train lab personnel, and even assist with troubleshooting of automated workflows. When a user's instrument is networked, we can also provide remote support for them. As with development, we work collaboratively with vendors to support our customers, and this includes co-traveling with vendors to customer sites. And overall, our wide range of experience across a number of platforms enables us to be able to provide automation, consultation, and support to both our vendors and our customers. So as a team, our goal is to develop methods that support the majority of our automation customers' needs, offering seamless implementation with minimal optimization. In order to do this, we follow a few core principles when developing our NGS workflows. The first, which we've touched on slightly so far, is that we focus on developing standard solutions. And this means that we design our methods based on the vendor's standardized NGS deck layouts, which allows us to reach more end users. As workflows and applications evolve over time, we're constantly improving our methods based on feedback from our customers and our field scientists in order to ultimately meet more of our users' needs. Next, we design our automated solutions to be modular. Designing methods modularly serves a few main purposes. The first is that it provides users with flexibility, accommodating workflow and lab setup differences. For example, many of our users choose to split their workflows across pre and post PCR lab instrumentation. By designing modular methods, we enable them to be able to do this without having to redesign the automated solution. Modularity can also provide crash recovery, enabling users to restart from any of the major points in the workflow as needed. And it also allows users to break up the workflow at the designated safe stopping points and restart when convenient to them. So the third core principle that we follow in developing methods is to include variable workflow options. Our kits and workflows are designed to support a range of applications. So we design our automated workflows to support that same wide range of applications with a single method. So for those users who have projects that span multiple application areas or sample types, they may need to be able to optimize their automated workflow on a project-by-project or case-by-case basis. And by providing variable workflow options upfront, users can test these variable workflow options themselves at runtime without having to edit the method. By designing intuitive user interfaces, we minimize the need for method-specific training, making it easier for our users to be able to train others in their labs on how to operate the automated workflow themselves. And lastly, for those instances where a method may need to be modified, we create easy to understand variables that allow trained users to modify methods with confidence and easily identify what a method is doing without needing to be a computer programmer. And so now that we've covered our approach to method development from a high-level perspective, I'd like to provide a closer look at the main stages of our development process and share more about what happens in each stage. So whether development projects are internal or collaborative, we generally follow the same process prior to releasing an automated solution to the field. So first, we would start with scoping. And during this stage, we define the workflow options based on the system's components and the user guide for the intended library preparation kit. So here we start to design the user interface along with the options that will be presented to the user at runtime. So for example, in this stage, we would define things like the minimum and maximum number of samples to run based on the system's deck capacity and whether those samples can be run in any format or if they must be limited to batches such as column sets. In this stage, we would also consider what cleanup options will be presented to the user at runtime, including whether we can present the cleanup ratios to the user to modify, or if the workflow requires specific cleanup ratios that should not be modifiable at runtime. So next, we would dive into the development and scripting stage using the vendor's provided software to write the method itself. And if the vendor has a standard method framework or design for NGS methods, we would follow that framework that's defined. So we make sure to include all workflow options supported by the kit's technical data sheet or user's guide. And we do try to write those easy to understand variable names, grouping them towards the top of the method so that they can be easily found and modified as needed. Some platform software functionality does include the ability to simulate a live run. So after we believe the script to be complete, we would be sure to simulate all options included in the standard workflow so that we don't get stuck troubleshooting script bugs later when we get to wet testing. So after the scripting is complete, we would move on to the optimization stage. And this is where we focus the majority of our time during development. Here we will do dry testing, water testing, and expired reagent testing to ensure accurate transfers of all reagents are made and the pipetting looks as clean as possible. And once the method is performing well with expired reagents, we would then move on to method demonstration, where we verify the performance of the method by testing with control samples and live reagents. We typically perform a side-by-side comparison with both automated and manually processed samples, which will go through various QC points and even sequencing to ensure comparable performance. And lastly, once we've demonstrated the method performance, we would use the data that's generated to publish an application or technical note that we can then share with our customers. So an important piece to our method development process is the demonstration of method performance and how we build confidence that a method is performing well enough to be released to the field for use with our kits. Our method demonstration experiments are designed to answer 4 main questions. The first question is, does the method perform comparably to manual sample prep? The second question is, does the method yield consistent metrics across a range of inputs? The third question is, does the method introduce cross-contamination between wells? And the last question is, does the method mitigate edge effects and plate effects for higher throughput runs? So we first want to show that the automated method performs as well as manual sample prep across a range of inputs as it relates to both library QC and sequencing metrics. When demonstrating performance of automated NGS workflows, we like to check the final library yields using our KAPA library quantification kit, and we will measure final library size by electrophoresis. We have an Agilent bioanalyzer in our lab, but we also have experience using other devices that perform similarly, such as the tape station, the LabTip GX-Etch, and the fragment analyzer. So if our QC metrics are comparable across manual and automated sample preps, we would then proceed on to sequencing. So now I'll review some of the sequencing metrics that we use, especially when demonstrating the performance of our RNA HyperPrep workflows, whether that's with mRNA capture, riboerase, or without an upfront enrichment option. So first, we would start with % map to genome. This is used as a global indicator of the overall sequencing accuracy and to check for the presence of contamination. Next, we would use duplication rate, for which we generally expect to see less than 10% duplicate reads. Duplication rate is a great indicator of whether the automated method performs as efficiently as a manual prep process, showing that the libraries created using the automated workflow are as diverse as manually processed samples. If we perform an upfront enrichment using mRNA capture or riboerase, we would then look at the % of ribosomal RNA reads to ensure an efficient enrichment process with minimal rRNA carryover into library prep. We also take a look at the general mapping rates between exonic, intronic, and intragenic reads. And depending on the enrichment process, the run reads are expected to distribute slightly differently. So for example, mRNA enrichment intentionally biases the read distribution towards exonic or protein coding reads. Lastly, we would look at mean coverage uniformity, the number of unique transcripts detected, and the number of genes detected, again, ensuring that the manual and automated libraries perform comparably. So we would next want to show that the automated method does not introduce cross-contamination between the wells. And in order to do this, we use negative controls that we place strategically throughout the sample plate. These wells will go through the entire library prep process without any template or initial starting sample. We would then QC these wells at the end of library prep to check for any detectable library molecules that might have come through from neighboring wells. It's especially important to check for cross-contamination in methods that may utilize shaking or multi-dispensing strategies. And lastly, our method demonstration should show that the sample wells perform comparably across the plate, especially for those higher throughput runs. So this means that when we run QC and sequencing, we would make sure to compare wells from various positions in the plate to ensure the automated method is not introducing edge or plate effects. So to help us answer these questions, our method demonstration experiments are usually run in two parts. For the first part, we perform a low throughput run where we run automated and manual samples side by side using the same reagent kit. We typically only include a limited number of samples for this run, anywhere from 8 to 24, depending on the platform, and we use control samples at variable inputs. When testing our KAPA RNA HyperPrep kits, we use universal human reference RNA as the control sample. And for the low throughput run here, we're really focused on assessing method performance at those variable inputs. So we would use the minimum and the maximum inputs that are defined by the kit's technical data sheet or the user's guide, as well as a mid-range input. So here's an example plate layout for our KAPA mRNA HyperPrep workflow, where the minimum input that's tested is 50 nanograms. The mid-range input that's tested is 250 nanograms, and the maximum input that's tested is 1,000 nanograms. So while testing input amounts in a low throughput run, we do use the same fragmentation conditions for all of the samples. And then we're sure to follow the TDS guidelines for things like the adapter stock concentration and the PCR cycle number. So this means that the 3 inputs will need to be cycled at 3 different conditions. So at the end of this experiment, we would compare the final library yields and sizes between manual and automated libraries. And if the libraries perform comparably across workflows, we would then move on to sequencing, selecting a subset of libraries across both automated and manual preps to sequence together. For sequencing analysis, we would then bioinformatically downsample or subsample the raw reads to an equivalent number in order to assess the final sequencing metrics across the 2 workflows. So assuming that everything performs comparably, we would then move on to the second part of our demonstration plan. And the second half of our demonstration plan does include a high throughput run. So in this experiment, we're looking for consistent performance across the plate. And so we run automated preps only at the mid-range input that was selected in the first experiment. We would then compare the final library metrics back to those of those same mid-range inputs that were run in the first experiment. So during this run, we would run the maximum number of samples that the method is designed for, which is typically anywhere from 48 to 96 libraries, depending on the platform's deck capacity. And again, we use control input samples. So in the case of KAPA RNA HyperPrep, those are universal human reference RNA. And we do include no template negative controls, which are shown in white on the plate. We usually will select about 10 to 15% of the total wells to run to be the negative controls, and we'll strategically place them throughout the plate. And this plate here shows a layout for a full 96 well run of our KAPA mRNA HyperPrep workflow. And as all of the inputs are the same, all of the other library prep parameters will also remain consistent across samples. So those include the fragmentation parameters, the adapter stock concentration, and even the PCR cycle number. If the platform that we're working on does have an on-deck thermal cycler, we would opt to use this device for all of the incubations. Otherwise, if the platform is not equipped with an ODTC, we would then use our standard off-deck thermal cyclers. And for this higher throughput run, we typically choose to spot check wells for QC. So shown here, the orange wells would be selected for QC, and these wells do include both wells with sample and negative control wells to check for that cross-contamination. Wells that are selected for QC will be checked for their final library yield and size, again, comparing them back to the first experiment, automated and manual samples for that mid-range input. If these do align, we would select a subset of these final libraries for sequencing. And for sequencing analysis, we would bioinformatically downsample or subsample the raw reads to an equivalent number that allows us to compare the metrics back to the first experiment. So at this point, assuming all of our metrics are comparable, we have effectively demonstrated this method's performance. And after documentation, it will be released for field implementation, either by our automation FASs or our vendor partners. So now we'd like to take you through a few examples of methods that are currently available on 3 different platforms for our KAPA RNA HyperPrep kits. And for each example, I'll start by giving a brief overview of the platform and its features. I'll then go into the method design and its features, along with any published documentation that we have for that method. So first up, we have PerkinElmer's Sciclone G3 NGS Workstation. This system's main features include a multi-channel head that can pick up individual tips, rows, or columns, along with an integrated gripper for moving labware around the deck. The system does come with a sunken deck option to increase tip storage capacity on deck, and it has 3 static Peltier positions along with a shaking Peltier position that can all actively heat and cool. The deck includes 2 weigh chutes, one that's mainly used for disposing of tips, while the other off the side of the instrument is mainly used for disposal of other labware such as plates and empty tip boxes. And finally, one of the main advantages of this platform is that it comes in a standard NGS deck layout. And that means that almost all of the instruments in the field are identical, which makes it very easy for the vendor and the vendor partners to develop methods that can be more easily distributed to customers in the field, as they won't require custom modifications based on the deck layout. All versions of our KAPA RNA HyperPrep workflow are readily available on the PerkinElmer Sciclone. The methods on this platform are broken into 3 main parts. The first is enrichment, the second is library preparation, and the third is the post-PCR cleanup. There are 2 separate method options for enrichment, which include both mRNA capture and riboerase. And at the end of both of these methods, the final samples are eluted and the enriched RNA samples are ready to directly proceed into the library prep method. On the left-hand side of the screen, you can see a screenshot of the user interface for our KAPA RNA HyperPrep method. Users are first asked to select what their initial sample type is, whether that's rRNA depleted or riboerase samples, poly-A enriched or mRNA capture samples, or other RNA samples if neither enrichment option was performed. The user is given the choice as to whether the adapters are provided are already arrayed into a 96-well plate, or if the user wishes to put these adapters into the reagent plate and have the instrument array them into a 96-well plate itself. And finally, the user has the ability to modify the 2 post-ligation cleanup ratios, although the recommended ratios are defaulted and they're presented to the user at runtime. So this method is designed to run in column sets, and the number of columns to run is actually controlled by an accessory workbook. The user does get a confirmation of the number of columns selected in the accessory workbook prior to the method starting, and that's shown in the image at the top left side of the slide. Finally, this platform does not include an on-deck thermal cycler, so all of the incubations will be expected to be performed in an off-deck thermal cycler. And so after the user selects the relevant options in the user interface, they would be provided with step-by-step instructions on how to set up the deck, including pictures of specific labware. So the deck setup instructions detail the specific labware required to run, including various Peltier adapters, magnets, consumables, and reagents. For detailed reagent preparation instructions, the user is provided an accessory Excel workbook that indicates, based on the number of columns selected, various volumes of reagents. It does include master mix formulation and specific consumables in which reagents are expected to be stored on deck. So once all of the reagents have been prepared and the deck has been set up, the user confirms that all of the items are placed on the deck and the method will start. And these methods do have an application note to go along with them that's published by PerkinElmer. In this piece, you will find an introduction to our KAPA RNA HyperPrep kits and the automated workflow, detailed instructions on how to initiate each protocol using the Sciclone, some highlights of the method features, including the user interface options and the deck setup instructions, recommended QC points throughout the automated workflow, along with a very small demonstration dataset. This method was developed collaboratively between PerkinElmer and Roche. So if you have any questions on the method or its features specifically, please feel free to contact us or your PerkinElmer representatives. And lastly, this method is owned and distributed by PerkinElmer. So if you would like to gain access to it, make sure to reach out to your PerkinElmer representatives. So the next example that we'd like to share is on the Tecan Freedom Evo NGS Workstation. And this system includes an 8-channel pipetting arm rather than a multi-channel head, along with a gripper for moving plates and labware throughout the deck. This platform comes with a tip storage shell for increased tip storage capacity, and it has 3 static Peltier positions along with a shaking Peltier that can all actively heat and cool. It does include a waste chute off the front of the instrument for tip disposal, and while Tecan offers many other versions of the Evo platform, our methods are designed to suit their standard NGS-seq layout, and that's what's pictured here. So we do also have all of our KAPA RNA HyperPrep workflows available on this platform. As the deck capacity is slightly smaller, the methods are limited to a maximum throughput of 48 samples per run, but the user is not limited to running in only column sets, and they can run anywhere from 1 to 48 samples. The workflow is broken into 5 parts. The first part is enrichment. The second is cDNA synthesis. The third is for the second post-ligation cleanup. The fourth is for PCR setup, and the fifth is for the post-PCR cleanup. There are 2 separate method options for enrichment, and that includes both mRNA capture and riboerase. And both of these methods leave the user with enriched RNA that can then proceed directly into the cDNA synthesis method. So at the start of the cDNA synthesis method, the user is then asked which of the 2 enrichment options were previously run. And as with the Sciclone, this platform also does not include an on-deck thermal cycler, so all the incubations are expected to be performed in an off-deck thermal cycler. The user is prompted at the start of the run with information on when and how many user interventions they can expect throughout the method. The user prompts are very clear, and they provide the user with detailed instructions at every intervention point. The Tecan software also includes a run status that shows both the method start time and the expected end time, again, making it clear how long the method execution will take. After the method options and the number of samples to process has been selected, the user will then be presented with instructions for how to set up the instrument for runtime. An accessory workbook is provided, and it instructs users on how to prepare reagents, including master mix formulations, based on the number of samples to run. Step-by-step instructions are then provided to the user for how to set up the deck, including reagent and labware placement on deck. So once reagents are prepared and the deck is set up, the user will confirm the correct placement of all of the items, and the instrument will proceed with the run. This method was also collaboratively developed between Tecan and Roche, and we're currently working on a technical note that reviews this method and the demonstration dataset in detail with Tecan. Tecan is ultimately responsible for the implementation of this method in the field, so if you wish to gain access to it, please contact your local Tecan representatives, and if you have any questions, feel free to reach out to us. So our third and final example of automated RNA-seq library prep is on the Beckman Coulter Biomek i7 hybrid platform. This system has 2 arms, which includes a span 8 and a multi-channel head, along with 2 independent grippers that can rotate up to 360 degrees. Users can choose to include static and shaking Peltier options that can actively heat and cool, along with an optional integrated on-deck thermal cycler. Users can also choose to include an ambient temperature orbital shaker and an active multi-channel head wash station that can be used for tip washing. The system also comes with a passive span 8 wash station and 2 waste chutes that can be used for both tip disposal and labware disposal on either side of the deck. The system is not a standard NGS deck layout. However, we have worked closely with Beckman to identify certain hardware requirements in order to automate our KAPA RNA HyperPrep kits. So currently, only our KAPA mRNA HyperPrep workflow is available on the Biomek i7 hybrid platform. However, we are in the process of developing a method for our KAPA RNA HyperPrep with riboerase workflow, along with our KAPA RNA HyperPrep workflow without any enrichment options. So on the left-hand side of the screen, you can see a partial screenshot of the method option selector or the user interface. So shown here are the general options, and the user is first asked to select a workflow for which we've included 4 templates. The first template is the mRNA HyperPrep complete workflow, which will run the entire workflow from start to finish, including the upfront enrichment and the downstream library prep, and it finishes with final libraries. The second template option is mRNA capture only, for those users who may want to break up the workflow across multiple days. The third option is RNA HyperPrep only, for those users who have paused after enrichment and they want to restart the automated workflow with library prep. And the final template option is called Select Individual Workflow Options. And this allows the user total control over all of the workflow options that are presented in the user interface that I'll actually show on the next slide. This template option is mainly used for error recovery purposes, but it does provide added flexibility to the user who may wish to run options that are not included in any of the defined templates. And so after a template is selected, the user is then expected to enter the number of samples to run, and this method is compatible with anywhere from 1 to 96 samples. Finally, the user is asked whether they want to use their on-deck thermocycler for PCR. So for those who may not have an integrated ODTC, this option is not applicable, and the user will be prompted to run all of the incubations in an off-deck thermocycler. But for those who do have an ODTC, this would allow them to choose whether or not to run PCR on deck, making the workflow easily compatible with labs that designate separate areas and instruments for pre and post PCR work. So once the relevant general options have been selected, the workflow options down below will populate accordingly. And depending on the workflow template that's selected, the options presented below might be locked and not modifiable. But regardless of the template that's selected, the user will still have the ability to modify at the very least the fragmentation time and temperature, the adapter transfer option of whether to transfer adapters out of a plate or individual tubes, along with the adapter plate starting position, the PCR cycle number, and the final library volume at runtime. Other options presented are modifiable by choosing the Select Individual Workflow Options template. The KAPA mRNA HyperPrep method on the Biomek i7 does include the use of guided lab setup, which is a feature of the Biomek software, and this provides step-by-step instructions for how to prepare all reagents, including master mix formulations and volumes to add of other bulk reagents. Instructions also include specific labware types and their deck placement, and once the user confirms that all of the reagents have been prepared and the labware is appropriately placed on the deck, the run will then start. So recently we published an application note featuring the KAPA mRNA HyperPrep method on the Biomek i7 platform, and this piece does include an introduction to our KAPA mRNA HyperPrep kit, along with the automated workflow on the Biomek i7 platform, reviewing the system features and those hardware requirements. It highlights the method features, including the user interface options, along with the guided labware setup for reagent and deck prep. And it presents the demonstration data set, including both low and high throughput runs at variable inputs, showcasing both QC and sequencing metrics. So while we did collaborate with Beckman on this application note, the method was developed internally at our Wilmington, Massachusetts location. If you would like to learn more about the method requirements, have specific questions on this method, or would like to gain access to the method, please reach out to our team. If your system does meet all of the requirements, our Automation FAS team would be happy to come on site free of charge to implement this method on your system. And this is the last example that we have to share, so we'll now review a few key points presented today before taking any questions that you might have. So to start, automation of your NGS workflows can offer some great benefits, including increasing your throughput and reproducibility. And when looking to automate your NGS workflows, it's important to find kits that are automation friendly, such as our KAPA RNA HyperPrep kits. Our automation team is here to help you automate your Roche NGS workflows, and we have a number of already available automated solutions across popular third-party liquid handling platforms. And our team is focused on developing easy-to-use standard solutions that suit the majority of our customers' needs. And lastly, if you're looking for automation support of any kind, please don't hesitate to reach out to us. And with that, we bring this webinar on Automatable RNA-seq library prep to an end. We would like to let you know that if you have any questions on the material that's presented today, I'd be happy to take them during the live Q&A session. Otherwise, you can always reach out to our Roche Sequencing and Life Science Support and Applications team via email or phone. If you're outside of the US region, please feel free to visit our Roche Global webpage to get connected with your local representatives. Thank you.